Hello everybody. Welcome to the webinar today for easier and more affordable cell cycle analysis at your bench. In particular, we're going to be discussing using FCS Express Image Cytometry with the Life Technologies Tali Image-Based Cytometer. And specifically, we're going to be covering cell cycle analysis and modeling of cell cycle data, acquisition through the Tali, and analysis in FCS Express Image Cytometry. So today we have on the line Shula Jaron from Life Technologies, who's going to be presenting on acquiring data or acquiring cell cycle data and cell cycle assays with the Life Technologies Tali instrument. My name is Sean Burke. I'm going to be presenting from the Novo software side about cell cycle data analysis and modeling in FCS Express Image Cytometry. So I want to change the presentation over here to Shula. Shula is going to start us off today talking about acquiring data or acquiring images with the Life Technologies Tali instrument. Shula, if you'd like to take it from there.
Okay, thank you, Sean. I'm just going to check in with you, Sean. Can you see my screen?
Yes, everything looks perfect.
Okay. So welcome, everybody. Today, as Sean said, we are going to be talking about easier and more affordable cell cycle analysis at your bench. And so the agenda for today is I'm going to start off by talking a little bit just about cell cycle and about the traditional fluorescence methods of measurement of cell cycle. I'm going to go over a bit about optimizing your system for your cells and for the assay that you choose. And then I'm going to talk about how do we use image-based cytometry to measure cell cycle. So this is sort of a new and exciting area to be getting into with, for cell cycle, and allows an easier assay collection that you can do in your own lab. And then finally, we will turn it over to Sean to talk about how to improve your cell cycle analysis using some additional software tools, including modeling of cell cycle. So first of all, cell cycle, really the cell division cycle describes the growth and then division of an individual cell into two daughter cells. And there are stages that the cells go through in order to create two daughter cells. They grow their cells, the cell grows normally, and then as it's preparing for cell division, it starts to replicate its DNA, prepares for mitosis during the G2 phase of cell cycle, and then goes through a cycle of division in the M phase. And during these phases, we can actually determine where the cell is in its stage of cell growth by looking at the DNA content of the cell. And so during the early stages of cell growth, we expect there to be 2N, 2 copies of each genome in the cell. And then as it's going through replication, it starts to replicate those copies until it gets to the G2 phase where it has 4N copies. And we can plot that on a linear scale. And if we plot that on a linear scale, what we'd expect to see in a population of cells is that we have some amount of that population that is in this G1 phase, G0, G1 phase with 2 copies of the genome. And we have some cells that have already made their copies, and they're in the 4N stage. And then we've got a number of cells that are somewhere in between those with a variable copy number. And so this graph that you see on the right is the plot in linear phase of the dye fluorescence if you stain your DNA, and the number of cells that are in each phase of that cell. And in fact, very traditional methods of measuring cell cycle, we can use, I'm sorry, I'm starting to hear an echo of myself in there. Okay, thank you. We can use flow cytometry to measure large populations of cells to see in a population of cells how many cells are in each stage of the cell cycle. And this, what you see now on the right-hand side, is a very typical histogram of a population of cells that are in just normal cell growth. And so in fact you see something very similar to what I showed you on the previous slide where you saw the very sharp bands at each of the stages of cell cycle. And in this case there's some error that create these sort of wider bands that come from both the instrument itself but also just from the cells being measuring those populations. And so we can use flow cytometry to measure a population of cells and determine where they are. And we can do this using a variety of different DNA dyes. And those DNA dyes can be used to label DNA in either live or fixed cells. And they have various different binding modalities. We have some dyes that bind to the major groove of the DNA, some that bind to the minor groove. We've got some that intercalate in between the base pairs, and then even some that are just external binders. But really the point being that there are many choices for DNA binding dyes, and they can be used both in live and fixed cells. And so if we look at just both some traditional dyes that are used and also some newer dyes that can be used for DNA content analysis and cell cycle, there are a number of cells shown on the top here that are traditionally used to look at nucleic acids in fixed cells, which in this case these mean that these dyes do not cross the cell membrane unless the cells have been permeabilized. And those are really good dyes to use for endpoint analysis. So if you are just looking at how a certain environmental or drug effect has affected your cells and you want to just see it at a single point in time. There are also now a number of cell permeant or live cell DNA dyes that can cross the cell membrane and give you a more in-process look at how cells are behaving during the cell cycle, during treatment by a drug. And what you'd expect to see for using either the fixed cell or live cell dyes is very, very similar. And so your choice between using impermeant and permeant cell dyes really it has nothing to do with the analysis itself. So in this case, on the top we can see two very common fixed cell stains, propidium iodide and Cytox Advanced, that again show the very typical histogram that you would expect to see with the G0, G1 phase shown on the left side of the histogram, the S phase with the cells that are in the process of duplicating their DNA, and on the right-hand side, those G2M phase cells that have duplicated DNA already. And that's true for the propidium iodide and Cytox Advanced. On the bottom, we have two dyes that are typically used for live cell staining, DyeCycle green and DyeCycle orange. So why might you be interested in choosing a cell permeant dye as opposed to a cell impermeant dye? Well, one very important reason is to look at long-term effects of a particular drug treatment or environmental factor on the cells in their growth cycle. And in this case, you can take the cell permeant dyes and stain your cells, look at them by flow cytometry, sort those cells, and then in fact regrow those cells after sorting and determine how they behave. The dyes themselves don't affect the cell growth of those cells, and you can see here that we've sorted our cell population into G0, G1 on the left-hand side and G2M on the right-hand side, and we've then replated those cells after sorting and watched as they regrow 3 days after the sorting event. You can see that in both cases, the cells continue to grow post-sort. So then you can continue to look at effects of your treatment and additional treatments post initial assay. So there are a few factors that you would want to consider when selecting a particular dye and a cell system for looking at cell cycle. So the first thing is that cell concentration with the same load of dye actually will affect your histogram that is created in the assay and your ability then to separate out the different populations of cells. So these are the same cells, the same growth conditions in all cases, and in this case all we've done is increase the concentration across from left to right of the cells in the assay itself. And you can see that there is an optimal concentration of cells sort of right in the middle here. You can see an optimal concentration of cells that allows you to separate out the populations of your cells and of the different stages of the cell cycle in your cell population. And so this is something that's pretty critical to performing an assay and being able to understand what is going on. In addition, dye concentration is also critically important. And in this case, we start with a increase in dye concentration at the top, and we've decreased the dye concentration towards the bottom. And again, you can see that at a very high dye concentration, you get sort of smearing of all of your populations, and it's very hard to distinguish the different populations of cells. And even in the low concentration at the very bottom, you don't have as good a separation of your three populations. Whereas right in the middle, you have sort of this optimal concentration of dye. And so these are things to consider. In most of the protocols, most of the protocols that have been developed for these different dyes have some guidelines on where to start, but often you will still have to optimize for your particular cell system. A couple of other factors that are important are incubation temperature for each dye, and these are clearly called out in all protocols. So in this case, on the top, you can see that the Herbst dye works very well at 37 degrees and gives you a really nice staining of your cells that can be separated quite nicely on the histogram. Whereas the Vybrant DyeCycle green really needs to be incubated. That dye needs to be incubated at room temperature with the for the optimal conditions. And so it's really important that when you are using a dye with your cell system that you understand what the optimal temperature conditions. And in addition, each dye has an optimal incubation time for that dye. And so again, in this case, we're looking at DyeCycle Violet on the top versus DyeCycle Orange on the bottom. And DyeCycle Violet's optimal incubation time in this here at 37 degrees is 30 minutes. Whereas DyeCycle Orange on the bottom, its optimal incubation time is 60 minutes at room temperature. So these are all critical parameters that need to be understood and followed in order to get the most optimal cell cycle distribution that is easily analyzable. And finally, collecting enough cells so that you can create a good histogram is of critical importance, no matter what technique you're using. And so here again, we're using flow cytometry and looking just at the difference between collecting about 15 to 16,000 cell events versus a little bit less than 2,000 cell events on the right-hand side. And you can see that although the histogram is still understandable on the right-hand side, and you could probably still separate out into the different stages of the cell cycle on the right-hand side, you get a much cleaner much more easily interpretable histogram on the left-hand side just by collecting additional cell events. So everything that I've shown you up until now has been looking at how to collect cell cycle information in a very traditional manner, which traditionally has been done using flow cytometry. And it's something that we've done quite a lot in our lab, is using flow cytometry to look at drug treatment and how it affects cells during the replication cycle. But we wanted to know, really, is it possible to do this in a simpler, easier, and less expensive manner? Traditionally, flow cytometry has meant that, and certainly in our case, we have a flow cytometry core, and we have to book time on the instruments in that flow cytometry core. So that means that I have to plan well ahead, make sure that the time is available in the flow cytometry core. I have to prep my samples in my own lab, transport them to the flow cytometry core, and then there is a cost, of course, associated with every hour I spend on that flow cytometer. And then finally, I have to understand sort of both the complicated software that's on the flow cytometer, but plus I have to understand any additional software that I might need to use to analyze that cell cycle. And so we were asking the question, could we do this in a way that didn't involve all of those steps and costs that we typically use for flow cytometry? And we decided that that image cytometry could be potentially a great tool instead of the flow cytometer. And there happens to be a benchtop image cytometer that our company sells, and we decided to go ahead and use this particular image cytometer to address the need of cell cycle at your bench. So what is image cytometry? So really, image cytometry is a combination of the quantitative aspects of flow cytometry with kind of the visual qualitative aspects of a microscope. And we decided, in addition, that we would go with a very traditional method of cell cycle analysis using propydium iodide, which is a very, sort of the oldest dye used for cell cycle and very common, lots of very common protocols out there. And we felt that that would be the easiest for us to implement and understand what was happening. And just as a side note, there are many different applications that can be used, that image cytometry can be used for, but we really, in this case, wanted to focus on how can it be used to give us really good, reproducible, and data that is equivalent to the data that we would get off of a flow cytometer. We chose to create a method, or use a method, basically, that has already been created in the literature, and keep it as simple as possible with the cell cycle assay with propydium iodide, just your typical steps to stain your cells and then analyze them as you would if you were doing flow cytometry. We're just going to use the image cytometry method instead. Suffice it to say that the image cytometer itself is very simple to use. I won't go through all the steps, but in short, it was very simple as loading the stained cells in a slide, putting the slide in the instrument, and then pressing go. And at the end of it, we would end up with our results, our cell cycle results. How does this particular instrument work? In this case, the slide itself is put into the instrument. The instrument captures images in bright field, in green fluorescent channel, and a red fluorescent channel. The parameters of those channels are shown here. It counts the cells in the bright field channel, and then it looks in the fluorescent channels to determine if those cells are fluorescent in either the green or the red. And then after it spits out that information, you have some ability to both gate on the cells that are of a particular size that you're interested in, and you can threshold on the fluorescence to determine whether those cells are fluorescent or not. This particular instrument can count up to about 35,000 cells in its maximum concentration and field of images parameters. What you end up with once you have collected your, once you have put this slide in with the cells and pressed go, in the end you end up with a number of images of your cells shown here on the left in the different channels, and you can look at those bright field images and you can look at the fluorescent images individually. And you also have some statistical information about the cells that are in the different regions of the histogram. In this case, I just want to point out that for this particular set of experiments, we counted about 10,000 cells. So we tried to keep, as I had stated before, counting as many cells as possible is really important, and so we were always trying to count as at least 10,000 cells for this particular assay. You also end up with a histogram that has some thresholding marks on it, and so you can separate out the different populations of cells here. And so you can see the number of cells on the y-axis and the fluorescence intensity on the x-axis, and then these bars that are movable that guide the data table to tell you exactly what is in each of those populations. And this looks very similar, and indeed is very similar to the type of histogram that we were looking at from flow cytometry, where we could see that the first peak here is the G0, G1 phase, the second peak is the G2M phase, and then the cells that are in between are in the S phase. So we were happy that we could use a traditional method of cell cycle, DNA staining for cell cycle analysis, that we could collect data that looked very similar to the data that we were collecting from the flow cytometer, and that we were able to do this in a simple manner at our benchtop. So then we went on to look at how does drug treatment affect the histogram or the data that's coming out of the instrument, and does that reflect what we expect from the drug treatment. So we decided to use Nocodazole to arrest the cells in the G2M phase. And here we show 3 of our tubes where we have on the left-hand side cells that have not been treated with Nocodazole. So we expect those cells to be in just a typical population of cells in the cell cycle. We have in the middle cells that have been treated with 25 nanomolar of Nocodazole. And so we expect to see some shifting towards the right-hand side, or the G2M phase peak. And then 50, on the right-hand side, 50 nanomolar Nocodazole treatment. And so we expect more cells to be shifted into that G2M phase. And in fact, we quite clearly could see that effect on our cell cycle data that we've collected from the Image Cytometry instrument. Very nice increase in G2M phase as we increase the amount of Nocodazole. And so that was good news for us, that we were seeing a phenomenon that we expected to see. And so we wanted to take it a step further and look at how does that data, can we pull out further that data by analyzing it using modeling, and how does that modeling then compare to modeling that we would of the same cells measured by flow cytometry. We have in our lab used for a long time FCS Express for flow cytometry to analyze our flow cytometry data, and we decided to look at using the Image Cytometry FCS Express to analyze our image cytometry data. And we were very fortunate to have had to meet up with the guys from Novo and to create this collaboration and create a way for us to import the Tali data into the FCS Express for imaging. And so all of our analysis from here on is really using the FCS Express for imaging and the FCS Express for flow cytometry. So we took that same experiment, and now we reran it, but we split our samples, and we ran half of the sample through the flow cytometer and half the sample through the image-based cytometer, and looked at how does that data compare. So going from no Nocodazole to 50 nanomolar of Nocodazole, how does that data compare in terms of the analysis? And so you can see on the top here we have the flow cytometry data, and on the bottom we have the Tali image-based cytometry data, and those populations modeled. And you can see that it is very similar. In the data on the bottom, the cells in each population are very similar between the flow cytometry data and the image cytometry data. So, as I said before, image cytometry has many different uses, everything from viability to apoptosis to oxidative stress and yeast and algae, but really it turns out that as a tool for looking at cell cycle in a population in a quantitative manner at your benchtop, there really is no other simple, inexpensive, and easy way to do it at your benchtop. So thank you so much. I'm going to turn it over to Sean now to talk about the modeling and how that works with the Tali data.
Hi, Shula. Thanks so much. So I'd like to start today by telling you a little bit about Novo software and FCS Express. FCS Express was originally developed for flow cytometry software, and we've recently introduced an image cytometry version that's compatible with the Life Technologies Tali. Now I want you to understand the workflow a little bit as well, and as you can see here, that the image data generation and the acquisition of the images is all handled by the Life Technologies Tali. So Tali will come in and acquire the images. It will then also perform image segmentation, or figuring out where on an image a cell lies, and once that happens, the Tali will also extract feature information. This includes things like the size and the shape of the cell, the intensity of different parameters or different stains that are included on that cell. Once this is done, all of that information can be seamlessly exported and imported into FCS Express. Once the data comes to FCS Express, you can then analyze that data further. You can easily create reports. You can then also communicate and publish those results with your colleagues. Now importantly, for cell cycle data, FCS Express also utilizes the Multicycle plugin tool, which will allow you to model cell cycle data quickly and efficiently. And that's what we're going to show you a little bit about today. Now, just to show you how easy it is to get your data from Life Technologies Tali into FCS Express, I want to show you a template that the Life Technologies and Novo software teams have put together, something that you can download from either of our websites to quickly analyze your data. So all you have to do is open up this template that again is downloadable from the website, and we'll give you the information where to get this towards the end of the webinar. But in FCS Express, we can click Add a Data File to our data list, and we're going to come in here and choose one of the data files from the Nocodazole titration series. And when we choose that file, it's going to be loaded up into FCS Express. All of our plots are going to fill with the information from that data. And since we're doing cell cycle modeling of this data, a multicycle DNA plot has automatically been inserted here. And we're actually fitting the data from G1, S phase, G2, and any noise or background into this model, and providing a statistics summary table where we're showing you the percentage of cells, number of cells, and even the concentration of the cells in the particular sample you analyze from each of the cell cycling populations, including some instrument information. Now, another nice thing about FCS Express is that you have access to all of the images that you acquired on the Tali. So that means if we want to look at our Propedian iodide fluorescence channel, we can quickly switch to that, and we can even magnify this axis here to zoom in and look at the individual cells. And again, if you have the bright field channel, you can quickly flip back and forth between the bright field, the red, the green, whatever it is parameters you may have. Now, the other great thing about working with this data in FCS Express is that you can quickly create a marker on this template. You can convert that marker to what we call a gate, and now all of the cells that are located within that marker gate are going to be highlighted in a particular color, in this case red. And as we move that marker around, we can see how our analysis updates in real time. So this is one of the great benefits of using these templates. You can see how easy it is to load that data into FCS Express from the Life Technologies Tali. As simple as just bringing it into your data list and you're ready to go. So let's discuss a little bit more about why you would use FCS Express, and we're going to get away from this template a little bit, and we're going to show you how this template was actually built up, and how some of these statistics are generated, and exactly how the modeling works in FCS Express. So again, one of the benefits to using a cell cycle model is we're going to get more accurate measurements of cycling cells rather than using a standard histogram approach. Now, just to review real quickly, again, you remember that cells in G0 and G1 are going to be 2N. They're going to have a variable amount of DNA as they go through the S phase, and they should end up in G2 with having 4N, or twice as much DNA as a 2N cell. So this is represented in a histogram, as you can see here on the right, that 2N cells has a specific peak in fluorescent intensity. Then you have a variable amount of intensity in dye fluorescence for the S phase. And when they reach 4N, they have another peak in intensity, and this peak is generally twice as much or twice as intense linearly than the 2N peak. Now, what this data looks like when you actually analyze it, or when you actually look at that data within another piece of software or biological results, is that a 2N population will actually have a spread. You notice that there's actually a CV involved, and that this data is spread out. It's not just on a singular line, and that in the 4N population there's actually a spread of that data, and there's also a spread going on here with the S phase. Now, if you use a typical or a kind of standard histogram-based approach to look at this data, you're setting up discrete thresholds where you say, I am going to look at the number of cells that fall under this area and the number of cells that fall within this area. Now, with this particular plot that we're showing here, we've actually modeled this data, and what you see is that there's an overlap of cells from G1 to S phase. There's also an overlap of cells from S phase to G2, and then you have that working backwards as well, an overlap from S to G1 and an overlap from G2 to S. So mathematical modeling is going to allow you to look at the probability that a cell, as it's going through late S phase, is going to be in G2, or the probability that a cell in G1 is actually in S, and it's going to allow you to look at the overlapping sections of these histograms, or these curves, to more accurately analyze that data. And the way that we handle this, or we've handled modeling data typically with flow cytometry data and now image cytometry data in the past, again, is to bring that data into a piece of software and to use something called gates to remove cells from the analysis that we may not want to include in there. Now, if you look at this model on the right, you notice that I have the 2N and the 4N population labeled here with a straight line. But above the 4N population, we have 4 plus N. And what does that exactly mean? And we have less than 2N, or a 1N, a half N population. Now, in most cases, when you look at data, whether it be from flow or image cytometry data, you are going to see these type of effects. But in a lot of cases, these are artifacts. And let's switch over here and take a look at what can contribute to these results when you analyze cell cycle data. Now, when you're analyzing flow data, I'm going to talk a little bit about the flow cytometry data, because it is very similar to the image cytometry data, is that when you analyze this data, we typically set up gates so that we look at what we know is a cell. So in this sub 2N area here, where I've labeled it 1N question mark, what this represents, it could be pieces of cell, it could be debris in your analysis, it could be a whole lot of just kind of small things that are floating around that are not that intense in your sample. So what we do in flow cytometry is set a gate on what we know to be cells in our analysis based on the size and shape of them. And if we apply this gate to our flow cytometry data, notice how we're going to lose this population here, because now we're only looking at the cells we know that are actual intact whole cells for the analysis. Now, another artifact that you can run into with flow cytometry data is that cells can clump. And when cells clump as they pass a laser, you end up with populations out here in the 4 plus N area. So why does that happen? You notice here in this diagram, we have a laser, and we have these cells, these yellow dots are representing cells, and as they pass the laser, they give a certain signal intensity over time, and you can see that here, signal versus time. Now, if two cells pass the laser at the same time, if they're clumped together, and this does happen even though it's not a perfect world when you run flow cytometry data, two cells that are in G1 that are stuck together will actually have double the intensity of G1 and appear to be a G2 cell. If two G2 cells are stuck together and they pass that laser at the same time, they'll then appear to be a 4 plus N population. And you can have any combination of these things. G2 stuck to S phase, G1 stuck to S phase. You can have 2 or 3 cells stuck together at the same time. And this is generally going to account for a lot of the kind of artifacts you're seeing up here on the high end. So to remove those cells from analysis and flow cytometry, we create a gate on the area and the height of a cell, and if we apply that gate to this model, you notice that we're going to significantly clean up this model. We're now not accounting for these cells in our analysis, and our analysis is going to be done more accurately and correctly. So when you're working with image cytometry data, you'll notice here on the right we have image cytometry data from the Life Technologies Tali. Similar artifacts can occur. Now, what we've done in this histogram to show you some artifacts is we've set up this marker gate, and when I apply this marker gate to what we call a cell gallery, we're able to look at the cells that fall within or underneath this area. Now, one typical type of artifact that can occur with segmentation is you can see this first cell in our analysis, or this first segmentation. In this segmentation, there's actually 2 cells included, and that also happened here down with cell number 7, as you can see. Now, what we want to do is remove those cells from our analysis, because if we count 2 cells that fall within the same segmented area at the same time, you're going to have a similar artifact as cells clumping together with flow cytometry data. So I'm going to apply this gate to the data grid, and you notice that most of the cells that fall underneath our marker gate are actually cells that are stuck together. There's many cells here that there's 2 at a time, and you notice that they're in this 4N plus population. Now, a similar thing can happen if we look at the low end of our histogram here. And in this case, when we look at the cells that fall underneath this marker, a lot of these cells are actually cells that appear to be half of a cell from the segmentation. Maybe the segmentation only picked up a piece of that cell. And again, as we move this around, let's see if we can't grab this gate and move it a little bit further. We are going to be picking up half cells from the segmentation, as you can see here. So those are, again, artifacts that we want to remove from our analysis because we don't want to analyze half of a cell that may give us a false sub G1 positive. Now, another thing that can happen when you're imaging data is that cells can be overexposed. So you actually take an image of a cell, it's too bright to be properly measured, and this will happen typically on the very high range of our analysis. So if we select these cells that are way out here, you notice in our cell gallery that these cells are in fact greatly overexposed. Most of the segmentation area is pixels that are going to be around 255 in value. So these pixel values are way too high to be dealing with, and we should exclude them from our analysis. So with these three artifacts mentioned, how can we refine our analysis using gates to better model this data? And that's what we're going to show you right now. So what I've done on another page here is I've actually created a multi-cycle DNA cell cycle analysis plot in FCS Express for our untreated Nocodazole sample. And what we're going to do is show you some ways that using gates will clean up this analysis to remove these artifacts from the 4N plus population, to remove some of these artifacts from the low end of this as well. So the first thing I'm going to do is move this plot off of the page here a little bit. And again, we're working in FCS Express, and this is going to work just like PowerPoint. So if I want to insert an object like a new density plot, all I have to do is click on that density plot, draw it, and now we'll have a density plot that represents all the cells from this Nocodazole treated sample. So what I really want to look at here is perimeter versus circularity as our first parameter. And what I'm going to do is that you notice that there's lots of cells with a high perimeter here. Cells that fall outside the main population or density of cells. So you can notice here we have purple, which indicates a very high density of cells, which is probably most of the cells in our analysis. So I'm going to create a gate around this area that I know to be cells of interest, and we'll call these cells of interest. And when we do so, you see that that population equals 86% of all the cells in our analysis. And I also want to show you real quickly here, if we look at the gate statistics, is that in this total sample there's 13,000 cells about, and that within that gate there's 11,450 cells. So by creating this gate, we're not actually excluding so many cells from our analysis that our modeling will be imperfect. We still have greater than 10,000 cells. So by creating this gate, the cells of interest gate, what we can do is actually visually confirm that the cells that fall within this gate are the cells we want to use for our analysis. So what I'm going to do quickly here is insert a cell gallery. I'm going to make some quick changes here so that we can look at the individual cell cutouts from this analysis. And let's make sure we get the right ones here. And now you can see here are all the cells in our analysis, and cells like this, these doublets, are what we want to exclude. So by applying this gate, this cells of interest gate, to our cell gallery, you can now see in the cell gallery that the majority of cells that we're looking at are in fact single cells as determined by the segmentation. And if we apply this gate to our cell cycle model, you'll notice that the cells in the 4N plus area have now greatly been reduced because we're removing those cells from our analysis. We're only including the ones that fall within this dense area of cells. So another artifact that we want to take into account is the cells that fall on the low end here. The cells that are actually kind of half of cells or segmentation debris, something like that. So I'm going to insert a histogram here, and on this histogram we're going to create a marker, and we're going to convert this marker to a gate. And this is going to be small debris. Now what I want to show you here is that if we insert a gate view, we have that small debris gate created. If we apply it to our grid here, you can see that cells that fall within here are either some sort of debris where there is no cell being shown, there's a little piece of a cell here, maybe this was a piece of a G2 cell, and other kind of segmentation artifacts that we want to remove from our analysis. So what we can do is actually make this gate a small debris gate. We can exclude those cells from our analysis, as you see here. And we can make this small debris cells gate a child of the cells of interest gate. So that means that anytime we apply this small debris gate to another plot, we're only looking at the cells of interest and all cells that do not fall underneath this marker. So let me apply that small debris gate, and we'll apply it over here to our modeling. And you notice that we've cleaned up a lot of the cells on the high end and a lot of the cells on the low end of our artifacts. Now, the one last artifact that we recommended, that we discussed, was that you can also have cells that are overexposed, or cells that fall on the very high end of the histogram. So let's insert one more type of plot, a color dot plot. And on this color dot plot, let's look at red intensity versus circularity. And you notice that we do have a number of cells that are appearing on the very bright end of this red intensity gate, or this red intensity marker. So if we include, let's grab all those cells on the top, and we'll color these orange, and we'll call these overexposed. You can see that these cells are overexposed, and if we apply that overexposed gate down here to our data grid, we can see by eye that what we're choosing here is in fact overexposed cells that we would want to exclude from our analysis. So what do? We can open up this overexposed gate, exclude those cells from the analysis, make it a child of the small debris gate, and now when we apply that gate to our multicycle DNA analysis plot and to our histogram, we're actually going to remove those cells from the analysis. And as we move this gate, you can see that all of our plots are going to update in real time. So by setting up this gating hierarchy, we've actually accounted for 3 different artifacts. We're including cells that are going to be singlets, we're going to remove small debris, and we're also going to remove overexposed cells from our analysis. And in the end, as you can see here, I'm going to bring our multi-cycle plot front and center, that our final gating is giving us this very nice cell cycle analysis plot where we're very sure that the cells that we're interested in this plot are going to be cells that are actually should be included in our analysis. So the next step here is that we'd like to get some data from this multi-cycle cell cycle analysis plot. So to do so, we can simply right-click on the plot, we can choose statistics, and we have a suite of statistics for working with cell cycle data, and I'm going to insert a few of these and then we'll describe what they mean. But again, simply right-clicking and choosing these statistics tables, you can insert them anywhere in the layout. And you notice here that in our basic statistics, we're giving you the G1 means, CVs, and percentages of cells. Again, doing that for G2, S phase, and also giving you some other statistics like the G2, G1 ratio. We then also have a model summary table, or a model summary statistics table, where we have 6 different models that you can choose from in FCS Express for modeling cell cycle data. And you notice when we click on the Multicycle tab, these 6 models are available up here. And what this table is telling us is that since the diploid and %S numbers here for most of these models are very similar, that this data is going to fit any of these 5 models equally, except for this last model where we use fixed CV values, and that's going to fall outside of our kind of range of working with this data in that model. We also provide a basic interpretation of this data. So you can see that no abnormal DNA content is observed. We're giving you the percentage of S phase and G2 cells, and you have a notification here that your S phase confidence is good. And since your S phase confidence is good, that means you can trust this model and that the data is working properly. Now, what we want to talk about as well is how is this data actually modeled in FCS Express by Multicycle? What components fit into this modeling to give you these results? So you can see in this multicycle DNA plot that in our G1 population, we have certain red hash marks highlighted here. In our S phase, we have some up and down red hash marks highlighted. And in our G2, we have another diagonal series of them. So these are each of the G2, S, and G1 populations that are being included in this analysis. There's also a green line showing us the general fit, and this is all on top of our black outline of the raw data itself. Now, the very nice thing about these models and overlays and all these statistics, again, is if we move this gate, this cells of interest gate, our analysis is going to update in real time to reflect that change to the gating. So you can see here some of our populations have changed a little bit and our percentages have updated. So let's make this a little bit more extreme. And you can see how all of these values are going to update for us in real time and change as we move our modeling or move our gates for our modeling. So if you want to access the different parameters that are associated with this model, and let's talk a little bit about that, is you can right-click on this cell cycle plot and choose Format. And when doing so, we have a whole category here called DNA, in which you can come and figure out or see what is going on with this data modeling. Now, there's two major things or three major things that go into computing a cell cycle model in Multicycle or FCS Express. The first one is the actual model, is the CVs of the data that you're trying to model. So the CV in this case is going to be the spread of the data in G1 or G2. If you remember in our theoretical model, there actually was no spread, it was just a straight line. But again, in a biological sample, you will have a spread or a CV. And the lower that CV value is, the better it's going to fit the theoretical model. So in this case, the Tali has done a very good job on acquiring the data and giving us lots and lots of cells to work with, so our CVs fit this model quite well. So we don't have to make any changes to this. Now, the model will also take into account the mean values of the G1 and G2 populations, which are going to be indicated by the mean value of the area under the curve for each the G1 and the G2. And this information is going to be plugged into the ratio of the G2 to G1 population. Now, what this means is that in the theoretical model, a 4N population should actually have twice as much DNA as a G1 or a 2N population. So therefore, the G2 to G1 ratio in a perfect theoretical world is 2. 2 times 2 is 4. Now, what will actually happen, as you saw, Shula had mentioned that there's certain things that can happen when you actually analyze or when you acquire an image and treat samples. Things like the amount of time that a sample is left with a dye, the temperature that sample is incubated, the incubation time, all those things can cause the G1 and G2 to actually be closer together or to be further apart. Now, ideally, they would be at a distance of 2. You would have a G2 to G1 ratio of 2. But what happens biologically is that these values can be different. So what we've done for this particular data set is we've set that value to 1.6. But let's say it was actually theoretically 2. What that means is when we look at the model now, the model is not fitting this data perfectly, right? Our S phase is very drawn out, and now we are missing our G2 population altogether. So by adjusting this ratio to the data that we see, and by having the visual verification that the cells we want to analyze actually fall within this model, and you can see as I move this down to 1.7, we're actually starting to see a G2. We can empirically determine that using a value of 1.6 here, which is what we've done for this data set, results in the best model for this particular data. So this is just something that we put in there to make this data fit a little bit better. You can see how easy it is to actually come in and make these changes to any of these fit parameters. And by doing so, we've actually made this data fit perfectly, and we know that, again, by looking back at the images, that this is where it should be. Now, one common pitfall, or one thing that you should keep in mind, is that the further you get from a theoretical model, mathematical model for working with biological data, it means that the further you are from what is expected through that theory. So that means if you are ending up with a G2 to G1 ratio of 1 or 3 or 4, that means that your data probably isn't fitting the model accurately. In that case, you may want to go back and look at your techniques and what has gone into preparing that data. And again, if your CVs are very great, if you have CVs in the 10, 20, 30, higher than that range, there's a very large spread of that data, indicating that cells aren't nicely fitting into the G1 or G2 populations. So that should all be taken into account when you're working with this data. But again, if you have the visual confirmation of what those cells look like and you've modeled that data, you can quickly go back and get that information. Now, when you're working with any type of data or any type of samples, you're always going to have more than one sample, I would hope. Again, this is the untreated sample of a 3-part titration series. So if we want to analyze all of the data from that titration series, we don't have to do that one by one. We can very easily add this data to what we call our data list. So if we want to add our 25 nanomolar Nocodazole treatment and our 50 nanomolar Nocodazole treatment, we can quickly and easily pick those files, add them to our data list, and when we hit this next button, what you're going to see is that the data on all of these plots is going to update to the next file in our data list. So you can see that we now went to 25 nanomolar Nocodazole treatment, and that all of our data is automatically being fitted for us at the click of a button. So again, if we click next and we go to our 50 nanomolar Nocodazole treatment, you can see that the G2 phase is really starting to bank up here, and we can really just flip back and forth now to look at all these data samples. So whenever this data, or whenever these templates or your layouts are set up in FCS Express, it's as simple as loading new data into it, clicking next, and getting some analysis from those data files. Now, anytime if we want to export this data to take a look at it somewhere else, in FCS Express you can quickly and easily export this data to PowerPoint. So let's create an export to PowerPoint and open it up for this one particular sample. And what's going to happen is PowerPoint is actually opening up on my computer now, and it's actually opening up on a separate screen that you're not seeing yet. And you can notice when I bring it over, our analysis is now in PowerPoint. We can resize these objects, put them on posters, present them to our colleagues, and in that way you can create quick, easy reports for this analysis. Now again, we mentioned nobody never acquires just one sample in their analysis, and here we've done 3. What if you have 50 or 100? We have an option in FCS Express to set up batch actions. So if we wanted to export all of the data from all of our samples here, we can say save this on the desktop to a new file. You also notice that we have some options here to export all of these to PDF, any statistics to Excel, even to Microsoft Word. And once we've set that up and click run on the batch options, let's review that we have everything set correctly. It's going to quickly analyze all 3 of these data files. So here comes sample 1, and now you can see on page 2, here comes sample 2, and again on page 3, our third 50 nanomolar Nocodazole treated cell is appearing. We can quickly flip back and forth in PowerPoint now, see the differences in all the percentages, and have all this information summarized in one easy report. So again, what you're getting here is the ability to create these templates if you'd like, load your data quickly and easily into that, get all the powerful statistics, model that data, and quickly publish this to something like PowerPoint or Excel or Word so you can communicate these results as needed. So with that being said, I'd like to summarize a little bit about what we've seen today. And as we go through this summary, you can also feel free to start entering any questions you have about anything you've seen in the webinar today in that questions and answers box. And as I mentioned, FCS Express was built on the flow cytometry backbone, so all the most proven and powerful tools for that type of analysis are included to improve your image cytometry analysis. This includes the basic cell cycle analysis multicycle plugin that comes free with FCS Express Image Cytometry. And this is going to allow you to move from population-based analysis to single-cell analysis at the click of a button. That way you can confirm cell phenotypes to clean up your models a little bit, and more importantly, you can remove any artifacts from your models with the fully interactive analysis between plots of image. All the plotting, the data analysis, the mathematical modeling, and advanced report generation tools, they're all in one package now. You don't have to bring this out anywhere else to start publishing that data. It's all right here. And we also offer many resources to our customers, like tutorials and sample data sets, an online forum, and really great customer support so you can get up and running with your analysis in no time. I also wanted to mention again that Tali example data set that we showed in the beginning and instructions for importing are available on the Novo software and the Life Technologies website. You can go there and you can actually get this template. You can load your data into it, so you really don't have to do anything except bring that data into a data list. You have your images displayed there, you have the cell-by-cell galleries, and you even have some information about the assay and how to work with that data in FCS Express. You can see here there's some simple instructions included. Now, I also want to mention that you can get a free trial of FCS Express to work with your own Tali data in the software. All you have to do is visit the Novo software website, click on the FCS Express demo in our download section, and you'll be able to come in here and download your free demo and import your Tali data directly into FCS Express. So with that being said, I'd like to flip over the presentation back to Shula to give a summary about what we've seen in the Tali, and we'll continue on with some questions and answers.
Yes, I'm here.
Great. Let me flip this back to you real quickly.
Thank you. And I would just like to summarize by saying that if you have additional questions about the Tali Image-Based Cytometer instrument, you can visit this website, lifetechnologies.com/Tali, to get more information about that instrument. And to say that also that all of the instrument plus the assays that I've shown you today, all are originating from molecular probes, which has a long history of these types of cell-based assays. And that is what I have for a summary.
Great. Thanks, Shula. And one other thing that I want to stress about working with this data in FCS Express, again, you saw that we have these templates available to you from the Life Technologies Tali, and this makes it as simple as bringing your data out of the box into the template, and your cell cycle analysis is complete here in FCS Express. And then you can take that to make it as complex or as simple as you'd like from that point. So we're going to remain on the line to answer a few questions here that are still coming in. And if there's any questions, feel free to ask them in the questions box. One question that came up was why was the G2-G1 ratio actually 1.6 as opposed to being 2? So Shula had mentioned that sometimes when you work with experimental data, depending on the incubation times of dyes, the incubation temperature, the concentrations of dyes, that can change the ratio of G2 to G1. So this time with this particular data set we used 1.6 to help fit the data a little bit better. Again, as your experiments or your assays vary or the variables in those assays change, sometimes you will have to make certain adjustments as the data varies from the theoretical model. And just to make it clear that if your G2-G1 ratio was 2.0 as expected, you wouldn't have to do any further analysis or any further changes. It would have detected that that model fit is perfect right out of the box. And Shula, looks like a question came in. Can we use image-based cytometry on whole blood samples?
Hi, I'm sorry, I'm having trouble looking at the questions right now, but yes. So you can use image-based cytometry for whole blood. You would have to dilute the whole blood sample so that individual cells were visible on the image. But other than that, it counts erythrocytes and leukocytes just fine.
Great. It looks like earlier on, Shula, that there was a question about price. Is that something you'd wanted to comment on, or where maybe you could point people to where they can get that information?
Sure. Our list price in the United States is right around $15,000, but we have a demo program where customers can have a demo instrument in their lab for a short period of time to test it out and to see if it fits for their uses. So I would recommend that anybody who's interested to please just contact us directly.
All right, and are there any other questions that are coming through here? And Shula, there's a question, can you use the instrument for assays like annexin 5 staining?
Yes, and there are a number of assays that have been optimized specifically for the instrument, including an annexin 5 staining assay. So we've got a couple of apoptosis assays that can be used with the instrument.
Great. And we had a question about how well the model will work with samples with 2 cycles, such as a mixture of tumor and normal samples. So in FCS Express, there are many different cycles that Multicycle can detect, including greater than 1 cycle of analysis. I would recommend that you just download a demo of FCS Express and try the data in there. If your 2-cycle data fits the model, you should not have any problems modeling that data in the software.
And then I see a question here that says, can you use 2 different channels to identify various populations? So the instrument itself has 2 fluorescent channels and a bright field channel, and yes, you can use the 2 fluorescent channels for independent staining and at the same measure at the same time.
And another question about the 2 different channels to identify various populations. That's something you can certainly do in FCS Express Image Cytometry, just like you would do with Flow Cytometry. In this particular data set, we're actually only dealing with 1 channel of fluorescence, but you may have noticed when we were showing multiple parameters such as size, perimeter, and circularity, that we could work with many parameters at the same time. So that will go for any of the fluorescent channels that you have with the data as well. So if you have 2, maybe 3 fluorescent channels, you can certainly create as many different gating strategies or look at as many different parameters versus each other as you'd like.
Okay, and I see a couple of additional questions about the Tali. The first one is, why was the S phase narrower than the flow cytometry S phase So the flow cytometer has a very sensitive detector, which also has a very large dynamic range. The image-based cytometer is part of the reason why we can offer it at such a competitive price is because we are using cameras to capture the fluorescence information. And so the dynamic range is narrower than a flow cytometer, and that then narrows the S-phase distribution. So can this instrument be used for GFP-expressing cells? Absolutely. It can be used for either GFP-expressing or RFP-expressing cells, or a combination of both.
And Shula, can the instrument be used to see, to look at platelets?
That is something that I will have to check with some other experts here and get back to you on.
Great. Okay, and then if there aren't any other questions, we're going to be wrapping up with the webinar today. If you do have any questions after the fact, you can contact us at support@denovosoftware.com. You can also, again, visit the denovosoftware.com website. You can visit the Life Technologies website, and we'll be following up with everyone after the fact as well. So thanks everybody for joining us in the webinar today, and thanks again, Shula, for the presentation.
Thank you everybody for joining.
Take care everybody. Bye-bye.